Finally. Alright guys. I am technologically dumb. Okay. So hi, I'm the drunk phytologist, and today we're infecting tobacco plants. Woo. Okay, so this is not the sort of, kind you smoke. It's the kind we use for research. So they're very short, they're easy to grow, and these are ones which I want to image under the microscope, and I may stream later this week showing that and how we do that. But first, what we're going to do is we want to actually express fluorescent proteins, so they glow in the dark when you hit them with a laser. And so what you see here, if you don't know if you can see it, you see these guys right here. Okay, so they have been infiltrated. So basically infiltration means that I have forced liquid into the leaf of a bacteria called agrobacterium, which will transfer the genetic material of my proteins into the leaf, and once they get expressed, they'll glow in the dark. So this can help us figure out where a protein is and what it's doing. And so right now I've done the first set. So this set is my protein just alone by itself, and we're going to look at that in about 48 hours-ish. And then here we'll go ahead and maybe see if we can share this with friends. Maybe not. We'll do it later. Okay. So these guys are going to be infiltrated with my favorite protein plus a protein from a pathogen. And what I want to see is if I have that protein plus my pathogen protein, if it is going to make a difference with their localization, so where they are, in the cell. Because it's possible that my pathogen is messing with my favorite protein. And that occurs because pathogens have a tendency to hijack this system. This happens in plants, just like it happens in people. And we kind of want to know how they do that a little bit. There's a lot of things that we don't necessarily know of how pathogens do this and are successful. And here, I'll give you just a second, real quick. Be right back. Alrighty, I'm back. Okay. So I'm just making sure my hands are nice and clean. And so, actually how we do this is we use a syringe, okay? It's actually no needle. And the reason is we don't need it. I have my two different bacteria. I'm going to hold on to my proteins. And so what I do is I take the bacteria up in the syringe, like so. And then I grab my leaf. You see my leaf here? And what we're going to do is we're going to force the liquid into the bottom part of the leaf. So this is where the stomata are. So the stomata are holes in the bottom of the leaf through which the plant breathes. And so we're actually going to be putting the liquid of the bacteria into it. Okay? So what I do is I put it like that. And then I just very gently push the liquid in. So I use just a little bit of pressure with my finger on the back side of it. And so now you can see here that liquid, sort of a different color in the leaf. And so that's the liquid that's gone into the leaf, and it's maybe a little bit easier to see from this direction. Okay. So what we can do is we put this bacteria into the leaf. It sort of fills up and goes in the layers in between the cell. So it kind of works like you do if you get like a shot or something like that, where it will go into your bloodstream, like underneath the skin. So plants have several different layers of cells. And so there's sort of the top layer, which is the epidermal cells. So epidermis, just like yours, skin, they're sort of their version of the skin. And then there are the mesophyll cells, which are underneath. And so they help with water fill, water exchange, all that fun stuff. And so this bacteria is going in between those layers, in between the epidermal layer and in between the mesophyll layer. And so the bacteria are going to infect this leaf and are going to help me what we call transiently express. So it's going to be, our protein is going to express for a little while, and I can capture that before the immune system of the plant gets rid of it, right? So plants have immune systems just like we do. However, their immune systems are, so I should probably say they have immune systems. Their immune systems don't work the same as ours do, but they can defend themselves. I know it looks like most of the time, since plants don't move anywhere, that they're like helpless, and they really aren't. And so we may talk a little bit more about that on a future date. But for today, we're just going to let our tobacco leaves sort of hang out. After this infiltration, I'm just going to leave them alone. Woo! Little bacteria went everywhere. Don't worry about me. This bacteria is a plant pathogen. It is harmless to people. So if I get some on me, it's not a big deal. Actually, I find it kind of pleasant smelling. It's very earthy, because this bacteria I'm using is a soil bacteria. So you'll find it pretty much anywhere. But we'll go over a little bit more about why I use this bacteria and how. And so there we go. Infiltration's done. So now these leavies, see where I put the syringe, are going to be waiting on the bench here in about 48 hours, and then we're going to look at them on the microscope and see if we see our protein. So that's going to be fun, and I'll show you guys some of the results later. So thank you for the first daily dose of science streaming. I am usually on Twitter at Everyday Science 2, so the 2 instead of an E on the very end. And we talk about all sorts of things on different days. So I will be trying to put out another blog post tomorrow about microbes. And so check that out at tinyfriendstinyfoes.wordpress.com. So thanks a bunch, everybody, and have a great day. Bye.